Hi, everyone. Welcome to DNALC Live. This is Amanda, and I'm at the DNA Learning Center today to present you with part one of a two-part series on the purification of green fluorescent protein. Green fluorescent protein was originally isolated from a Pacific jellyfish back in 1962 called Aequoria victoria. I'm going to zoom in on the picture of this jellyfish. Turns out this jellyfish protein, green fluorescent protein, has become an indispensable tool in the field of molecular biology and genetics research. I'm going to tell you the story of how GFP was discovered, how it's used in research, and walk you through a laboratory technique to get bacteria cells to make this jellyfish protein in batches so that we can isolate and purify it. Why do you think this jellyfish glows? So it turns out they don't glow all the time, but if something bumps into the outside surface of the jellyfish, so let's say, I don't know, there's a hungry fish swimming around and it sees the jellyfish and it boop, bumps into the jellyfish lights up very quickly. So if you were the hungry fish and you bumped into a jellyfish and it illuminated itself quickly, what would you do? Yeah, I'd do the same thing. I'd swim away. What scientists think is that Aequoria victoria uses this trait of fluorescence to protect itself from predators, to startle them. And it turns out this trait is genetic. Genes are pieces of DNA. Oh, pardon me. And they're pieces of DNA that are like recipes. They carry coded instructions that ribosomes inside the cells of all living things, from bacteria to humans, can use to make proteins. So I'm going to make a little schematic here that you may have seen before. Genes are recipes that tell our cells how to make proteins. And what do these proteins do that our cells make? Proteins ultimately will play a role in the determination of a trait, a genetic trait. So in humans, can you think of an example of genetic traits? Eye color comes to my mind. So eye color is determined by melanin proteins. So you inherited genes that tell the cells in the iris of your eye to make beautiful proteins, and as a result, you have a particular color in your eyes. Well, it works the same in the jellyfish. There is a gene that tells the jellyfish cells to make a protein called green fluorescent protein. That's a long one. And that protein glows inside the jellyfish cells. Pretty cool. The gene has a name. It's called the GFP gene. And GFP just stands for green fluorescent protein. So let's go back to the beginning. There was a scientist in Japan in 1962 who actually was the first to publish an article about this protein. His name was Osamu Shimomura, and he shared a Nobel Prize in 2008 with two other scientists, Martin Chalfie and Robert Sien, for the discovery and development of the green fluorescent protein. Dr. Shimomura was a marine biologist. Little did he know that years later he'd be given a prize for his contribution to research that affected molecular biology and genetics. But he first isolated the protein in 1962 from this particular jellyfish. Years later, Martin Chalfie, who works at Columbia University here in New York, was the first to show that that protein from a jellyfish could be used as a genetic tag or marker. On the screen, you'll see a picture from the cover of a journal called Science. And if you can't tell what that is on the cover, it's a picture of a little microscopic roundworm called C. elegans. And that was the model organism, and still is, that Martin Chalfie uses in his research. So what does it mean to use GFP as a genetic tag or marker? I'll show you some pictures. A lot of times in molecular biology, what scientists are trying to understand is how genes code for proteins and what those proteins do. So imagine you have a gene of interest. You can see it in red on my screen. And the arrow leads to the protein. That's the three-dimensional structure of the protein. It's big, long ribbon strands of amino acids that has a function inside a cell. And you want to better understand when that protein's being made, where in the cell it's going, if it's being transported to other parts of the body. Well, in order to see that protein, which often is invisible, what you can do is you can tag it. What that means is you can insert the GFP gene into the DNA of the organism that you're studying, right beside the gene for the protein that you're studying. And anytime your gene of interest is being expressed, meaning it's being used to make the protein you're studying, it will be attached to the GFP protein, and it will fluoresce. So everywhere your protein goes, it will glow. You can see where it is when it's active, and you'll know when your gene of interest is active in the life of a cell. If that is a gene that causes a disease, you'll know when that disease gene is active. Very, very useful. So often the GFP gene is called a reporter. It reports back to you, meaning it makes a protein that you can see. You know when the gene is being expressed just by looking at the cells. Here's a picture of some mice that were genetically engineered with the GFP gene. So you'll see 3 little pups here that are bright green and 3 in the middle that are not. They look a little bit like baked potatoes to me. The 3 genetically engineered mice are clearly different than the mice in the middle that I would call the control mice, no new genes. And I know that if my goal was to make mice that glowed, that my experiment worked. And I know that it worked because those mice now glow. The gene is telling me it's there. Makes a lot of sense, right? Why would a scientist make a glowing mouse? My guess is that this was practice. If we can insert a new gene into the cells of a mouse, odds are we could insert a gene into the cells of a human. Does that mean that someday our goal is to make glowing people? I think not. My guess is that it has to do with human health. Maybe if there are genes inside a human cells that are malfunctioning and causing a disorder or a disease, they could be replaced with healthy human genes. So this technique is genetic engineering. We'll be talking more about that later. The third scientist who contributed to this Nobel Prize winning work with GFP is Roger Tsien. He unfortunately passed away a few years ago, but in his lab in California, the Tsien lab, they made variants of GFP. What does that mean? Well, if you look beside his photograph, there's a picture of a petri dish with bacteria growing in it. All the letters and dots you see are globs of bacteria growing in a dish, and you'll see they're not just green. There's orange, there's yellow, there's red. Those are all variants, meaning the protein that's being produced is slightly different, and as a result, it glows a different color. Simply by changing the recipe in the gene, you can change the protein slightly and then change the color that's being expressed. And here's a really cool picture of some little tubes of bacteria in a rainbow of colors created at the Tsien lab, and then a beautiful picture of a seaside landscape that was all drawn with fluorescent bacteria cells, cells that were genetically engineered with these interesting variants of GFP. Now, how is this useful? Well, let me show you this picture. This is a famous picture called the brain bow. In this picture, we're looking at neurons or brain cells. They're all clumped together, but they're different colors. And how is that helpful? It's very easy to tell one cell from the next and to trace cells that are related to each other. Having lots of different fluorescent tags allows us to see lots of different cells in the same tissue. Very useful. So in our experiment today, what we're going to try to do is get a lot of this green fluorescent protein from some bacteria cells. If we want a lot of a protein, we have to program cells to make the protein for us. And that's where genetic engineering comes in. We have the ability to take a gene and to insert it into the cells of other living things. And it turns out that bacteria are an organism that are used in the production of lots of proteins, bulk production. I mean, you can grow vats of bacteria, flasks of bacteria, and if they're genetically engineered with a human gene or a strawberry gene or a jellyfish gene, they'll make lots and lots of the protein that that gene codes for. So in our lab, what we're going to do is genetically engineer bacteria like these. This is an electron micrograph of E. coli cells. We'll talk more about them in a minute. To make lots and lots of green fluorescent protein. And then tomorrow you'll learn how to extract and purify the protein, which is a lot of fun. But it actually is a very practical technique. And it's used a lot in pharmaceutical labs. What happens at pharmaceutical companies? They make medicines, they make vaccines. Well, I'm going to show you a picture of a product that's made at pharmaceutical companies here called humulin. Humulin is human insulin. Human insulin that's made inside bacteria cells. So if you don't know what insulin is, it's a hormone secreted by the pancreas that can regulate blood sugar levels. In some forms of diabetes, patients have to actually supplement their bodies with insulin to regulate their blood sugars. So that insulin is put into a needle and injected into the body. The insulin's purchased from, the pharmacy. The pharmacy's getting it from pharmaceutical companies. Where's the pharmaceutical company getting it? They're not extracting it from humans. Decades ago, insulin used to actually be extracted from animals, like pigs. Not living animals, though, from organs that were left over in slaughterhouses where animals had died. You can extract and purify pig insulin, let's say, put it in bottles and give it to humans who need it, and it works. But some people have allergic reactions. It might not always be possible to completely purify the protein, so there's risk sometimes of tiny little virus particles sneaking through. Nowadays, here in the United States, the insulin that's sold, humulin is one of the product names, is actually human insulin. It's not pig insulin. But the difference is that it's not made in human cells, it's made by bacteria cells that were genetically engineered. If I can find the gene, the instructions to make human insulin, let's imagine that's the insulin gene, what I can do is I can glue that insulin gene into a little molecule called a plasmid, which is like a gene taxi. And I can take that whole plasmid, that little ring of DNA containing this gene of interest, insert it into cells, and they'll make the protein for me. So if you take insulin or you know someone who does, you can thank E. coli most likely for the production of that insulin. And there are a whole bunch of human proteins that are produced in this very same way by genetically engineering bacteria. Now, what's so great about bacteria? I'm sure that's what you're thinking. Most people hear the word bacteria and they think of yuck. They think of germs. I'm going to move my picture up here. So in this picture, we can see some of the really cool things that make bacteria special and unique and really good to become protein factories. Number one is their size. So I'm going to circle one individual organism in this pile. Boop. It's a single cell. It looks a little bit like one of those puffy cheese doodles to me. So bacteria are what we call unicellular organisms. They're single-celled, very, very small. Bacteria cells are far smaller than animal cells and plant cells. So you can fit a lot of them in a pretty small space. And what about their care? Do they need bathrooms, special food? No. You can put them in a tube with liquid food that has everything they need, or a flask, and they can sit on a shelf and they'll be quite happy. The second cool thing you can see in this picture is right here. I'll circle it again. What is going on here? It looks like it could be a vicious attack, but I don't think that's what it is. It looks like right down the middle here we have a line. What do you think is going to happen right at that little line? The cells are going to split. So what we're seeing here is the process of cell division. When you're only one cell big to reproduce, you split. You divide into two clones, two new identical cells. Bacteria reproduce through cell division, and they can do that very quickly, if they're happy. If you give them the food they like and the temperature they like and mimic the environment where they normally live in a laboratory, you can coerce them gently to reproduce every 20 minutes. They can double in number every 20 minutes. So if I start out with, let's say, one cell that has the gene to make insulin in it, and I give it food and keep it warm for 24 hours, that cell will start to divide, and then its daughters will divide and divide and divide, and in 24 hours, I can have billions of insulin-producing cells in a small tube. There's one more really cool thing about bacteria that we can't see in this picture, but you might be able to see it on my little cartoon picture here. Bacteria cells are what we call prokaryotic. Animal cells and plant cells are eukaryotic. Prokaryotes, like bacteria, are very simple in structure. So in my diagram of this E. coli cell, this stringy stuff in the middle, that's its DNA. It has one chromosome. These little dots are ribosomes. That's where proteins will be produced. And then the outside of the cell is surrounded by a membrane or a wall. That's about it. In an animal cell or a plant cell, there are lots more structures. And there's one major structure in prokaryotes that's quite notably missing. Oops, I didn't mean to do that. If you noticed it too, good for you. There's no nuclear membrane. The DNA is just floating around unprotected inside the cell. In an animal cell or a plant cell, there's DNA, and then it's protected by the nuclear membrane. That is clearly absent in prokaryotic cells. The reason this is good for our experiment, and you may have already figured this out, is that what we're going to try to do is genetically engineer bacteria cells. We're going to try to put new genes in that wouldn't normally be found there. We're literally going to float those new genes in through the surrounding membrane, and then we're done. If we're using animal cells or plant cells, those genes not only have to pass through the outer membrane or wall, they have to get into the nucleus too. That requires a whole extra set of steps. Engineering bacteria is very easy because they're so simple. So we now have an organism that's single-celled, so we only have to get the gene into one cell. It's prokaryotic, meaning I only have to get that gene through one barrier, and it also reproduces extremely fast. A perfect host if we're trying to make lots and lots and lots of a new protein. Now you'll see on the top of the board, I wrote the name of the bacteria. I already said it too. We're using E. coli for this experiment, which some people might find very unfortunate because most people think of what when they hear E. coli? Yep, they think of food poisoning. The E. coli we're using in our experiment are called MM294. MM stands for Matthew Meselson. He's the scientist who created this strain of E. coli. And this strain is a laboratory strain, meaning it can't make you sick. It's very robust and relatively easy to keep alive in a laboratory. It's genetically different than the ones that cause food poisoning, but it is related. This whole scenario reminds me of dogs. Does anyone have a pet dog at home? I have a few dogs, and I know that there are lots of dog breeds. They come in different shapes and sizes. They have different personalities. Well, in the E. coli world, there are lots of strains, which are like breeds of E. coli, and they vary one to the next. Some are friendly, and some not so much. Luckily, in our experiments here and in pharmaceutical companies, the E. coli that are used are very, very safe. So what I'm going to do now is I'm going to show you a very quick, easy way to genetically engineer bacteria. The one tricky thing is getting the genes into the cells themselves. I already mentioned a structure called a plasmid. These are the tools, we call them vectors, that scientists use to transport new genes into bacteria cells. And I'll tell you why. Plasmids are little rings of DNA that are normally found in bacteria. Yep, that's right. I'm going to draw a bacteria cell up here. E. coli have flagella. They have lots of little tails. Here's its chromosome. Here are its ribosomes. And often there are these plasmids, extra little pieces of DNA floating around inside the cells. Sometimes there are genes on these plasmids that will offer some kind of advantage to the bacteria cells should the environment change. Pretty neat. So bacteria don't mind having plasmids inside them. And what we can do in a genetic engineering experiment is literally, let's see if I can ungroup these things. Remove a plasmid from a bacteria cell. I'm going to make it bigger here. Whoa. And use special enzymes called restriction enzymes to create an opening. And into that opening, I can insert and ligate or glue any gene I want. The insulin gene, the GFP gene, doesn't matter. And then I can make sure that everything is glued together. And then I can insert that recombinant plasmid, I've recombined new DNA into it, into bacteria cells. And that bacteria cell doesn't mind that it's there. It's used to bacterial plasmids. And another cool thing is that when there's a recombinant plasmid introduced to a bacteria cell, guess what it does? It makes copies of that plasmid, lots of them. So if I'm a scientist and I made a really cool plasmid and I want to make more of those plasmids, all I have to do is put them into a cell and I can let the cell make the plasmids for me. And when this cell divides, those recombinant plasmids will be passed on to the offspring. So the plasmid that we're going to use in our green fluorescent protein purification lab is called PGFP. P stands for plasmid, and GFP is the gene of interest in our plasmid. So one section of the plasmid is the GFP gene. If a plasmid gets into a cell, that cell will have the ability to make green fluorescent protein. PGFP has a second gene in it, and this is an important part of our experiment that we'll discuss in a moment, called the AMP-R gene. This is a bacterial gene. And the trait that it's associated with is Ampicillin Resistance. Have you heard of ampicillin before? It sounds a lot like penicillin. Yep. Ampicillin is an antibiotic. So if you're resistant to ampicillin, that means you're a cell that can survive in the presence of a drug that would normally kill you. Antibiotics are drugs that kill bacteria or inhibit their growth, prevent them from being able to divide and survive. Ampicillin normally inhibits the growth of E. coli strain MM294. But if there's a gene like this AMP-R gene in E. coli cells, they will be able to live. Good thing? Bad thing? Let me go back a slide. So let's say you have an infection in your throat, not E. coli, but strep. If you've had strep throat, you know it's quite unpleasant. When you go to the doctor will do a strep test and maybe prescribe an antibiotic, depending on the results. If the strep bacteria in your throat are resistant to the medicine you're taking, what's going to happen? Nothing good. You're still going to feel sick. So it's actually not so good for us as humans when the bacteria that infect us are resistant to drugs. But who is it good for? It's very good for the bacteria. In nature, having genes like resistance to antibiotics genes is really helpful. In our experiment, the goal is not to make antibiotic-resistant bacteria. We want bacteria that are making green fluorescent protein so we can harvest it. But we use a plasmid that has both genes, and here's why. When we insert these plasmids into bacteria in a moment, only about 1% of the bacteria are going to get new DNA inside them. 1%. That's not a lot. There are going to be 99% of the cells left exactly the same as they were when the experiment started. No glowing. Now, if our goal is to extract and purify green fluorescent protein, I only want the cells that glow, that got new DNA inside them. So how do I get rid of the cells that do not have new DNA in them? the ones that were not successfully genetically engineered. I use an antibiotic. What we're going to do is we're going to culture our experiment cells in a petri dish that contains ampicillin. It's mixed into their food. Any cell that doesn't get new DNA in it will not be able to reproduce. Its growth will be inhibited. But cells that do get new DNA inside them are going to be able to live. This gene will allow them to survive on this very special, we call it selective petri dish. And in addition, if they are resistant to ampicillin and can survive, they are also going to glow. Pretty clever, right? So let's start our experiment. I actually have a beaker of ice here, and in the ice there's 2 tubes. So I'm going to come up closer to the camera to show you. One of the tubes is actually labeled PGFP. And if you look really closely in the bottom of the tube, do you see that tiny little bubble of liquid? That's 10 microliters of liquid. Microliters are really small volumes. How many plasmids, PGFP plasmids, are in that tiny little volume of liquid? A lot. Thousands and thousands and thousands. So we've got a lot of plasmids carrying that jellyfish gene in that tiny little bit of liquid in this green tube. My second tube is clear. And what you should see is that it's got a cloudy liquid in it. That's actually salt water. It's a calcium chloride saltwater solution. And salt water isn't usually cloudy, it's clear. This is cloudy because right before this lab started, I put some bacteria inside the tube. I'll show you where I got the bacteria from. They were living in a petri dish. And you see all those stripes and dots? That's all bacteria. Yeah. They've been living in a dish where there's food. They grow on the surface of their food. It's that yellow stuff called agar. I scraped some of the cells off the agar, and I mixed them very carefully into this saltwater solution. So it's nice and cloudy. How many cells do you think there are inside this tube? Here's my clue. I can see them. It's very cloudy. There's even some piled up in the bottom of the tube there. If they're visible, what does that mean? 1 cell? Absolutely not. There's hundreds of billions of cells inside this tube. Lots of host cells that hopefully will receive plasmids in a moment. Okay, so I'm going to go back to my workstation. I mentioned the unit we'll be measuring in. It's called microliters. Does anyone know what a microliter is? Micro means very small. Here's the abbreviation for a microliter. It's the Greek letter for M. We call it a mu. Maybe you've heard this abbreviation or used it in science class. ml, milliliter. They're related. A microliter is just a fraction of a milliliter, though. Maybe you've heard of liters, yeah? Milliliters are fractions of liters. So a microliter is 1/1000th of a milliliter. Now, milliliters are pretty small. You maybe have measured teaspoons of medicine. A teaspoon is 5 milliliters. That's 5,000 microliters. They're really tiny. The only way to measure microliters accurately is with an instrument called a micropipette. I've got one right here. I'm going to set my micropipette to 250 microliters. In this experiment, I want to transfer 250 microliters of bacteria in salt water. Can you see that? Into my tube of plasmid DNA. So I'm all set. And this works a lot like a transfer pipette or a dropper, which probably you've used before. The only difference is that because I use this piece of equipment for lots of experiments, I put disposable tips on the end. They're in boxes like this. I'll show you how it works. I just take my instrument and I press it into a tip and it sticks. It's very long and it's sterile. What does that mean? Just like a Band-Aid before you open it up. It's super clean, no germs. Should I touch it? Should I scratch that itch? No, keep it clean. The only bacteria I want in my experiment are the bacteria in this tube, my E. coli, strain MM294. So I'm going to open this tube up and I'm going to use my micropipette to transfer the bacteria out of this tube and into the plasmid tube. Before I put this pipette tip into the liquid, I always press air out. It's just like squeezing a dropper. I'm kind of turning the instrument into a vacuum. I hold my thumb down, put it into the liquid, and then I slowly release. And the liquid gets lifted up into the pipette tip. Here it is. So now there's 250 microliters of cloudy bacteria liquid in my pipette tip. I'm going to throw the extra bacteria into the trash. I don't need that. And I'm going to push the bacteria out now into the tube where my plasmids are. So before I press that button, I'm going to put the pipette tip all the way into this tube and then push the bacteria out till that button can't go any further. And I hold it down until I've removed the tip from the tube. I don't want to suck any of that bacteria back up. All right, it's all in my green tube. I can tap it to mix it. And now I've got bacteria and GFP plasmids in one place. I'm going to put this tube back into the ice. And here's something fun. This pipette tip, instead of using my fingers to remove it, there's this eject button. Yeah, very satisfying. Tips in the trash. All right, so what have I done? I've combined my host, the bacteria cells that I want to receive new DNA, with the plasmids that contain the genes that I want to get into the bacteria cells. How do I get the plasmids to go into the bacteria? So I've got a little diagram here. I'm going to zoom in so you can see this picture better. Here I've got a few little bacteria cells, got some plasmids floating around. This is what's going on inside my tube, and the tube is sitting on ice. It's very cold. Cold's important. Cold affects the movement of molecules. Can you tell me how or in what way cold affects the movement? I'm going to draw the outside of my tube while you're thinking. Yeah, when molecules get cold, they tend to slow down. My blue marker does not want to work today. There we go. So let's say this is my tube. I've got 2 giant bacteria cells and some plasmids. Multiply that by billions, we're good to go. Well, if everything's sitting very still, that's like the opposite of what we want, because we want movement, right? We want plasmids to be able to go into the bacteria cells. So how do I make everything move? Exactly. I use a little bit of heat. So I have the cells right now sitting on ice for a reason. We always start with cold cells. The next step to induce movement is to take that really, really cold tube and to dip it in water that's hot. So here's my hot water. Ooh, hot water, lots of heat energy. Specifically, I'm going to dip that really cold tube in water that's heated to 42 degrees Celsius. Oh, can you see that? I'm going to write it up here too. That's about 108 degrees Fahrenheit for all you Americans. And I'm going to heat the tube for 90 seconds. So what happens when I take a really cold tube and I dip it in hot water? Yeah, heat energy from out here is going to enter that tube, and it's going to enter the tube very quickly, like a very quick hot draft, it's called. And it happens very quickly because we made sure that inside the tube was very, very, very cold. So there's what we call a thermal gradient, a difference in the temperature inside the tube and outside the tube. That means heat will enter quickly. And when that happens, everything in the tube gets moved around and plasmids can get pushed into the bacteria cells. Have you ever felt a hot draft before? I have. When I go to the grocery store during the summer and it's really cold in the store from the air conditioning when I leave, the door is open. And when the door is open, all that hot air outside slips through the door into the cold grocery store from an area of high concentration. So there's the high concentration of heat to an area of low concentration. Same thing's happening in our experiment. The heat energy is just moving. It wants to fill the space. All right, let's say some plasmids get into some bacteria cells during this process of heating. How do I make them stay? Well, step 3 is making them cold again. If I make the tube cold very quickly after the heat step, the plasmids will stay. Everything will stop moving. This process of heating and cooling the cells is called a heat shock. Heat shock. We're going to shock the cells with a very quick, distinct change in temperature, and that's how we move the plasmid into the bacteria cells. So to shock the cells in the lab, what we do is keep the cells on ice until we're ready to start timing 90 seconds. And to heat, I have a water bath set up right here. It's at 42 degrees Celsius. So I remove my tube from the ice and I put it in the bath for 90 seconds. During the 90-second incubation, heat energy is entering that tube very quickly, and hopefully some of those cells are receiving new plasmid DNA, and they'll start to make some protein for us. Let's imagine 90 seconds has passed. When I remove the tube from the heat, I go directly back into the ice and try to stop the movement of the molecules quickly. So at this point, some of the cells inside my tube have been transformed. What that means is that plasmid DNA has been pushed inside them. They've been genetically engineered. Pretty easy. All I need to do now is give those transformed cells some time to multiply. How do I get them to multiply? What are the things that make bacteria happy so that they reproduce quickly? Do you remember? They need food, and these bacteria happen to like very warm temperatures, body temperature actually, which is 37 degrees Celsius. So what I'm going to do now is transfer the bacteria from my experiment into a nice clean Petri dish where there's food, and then they'll be incubated overnight at 37 degrees Celsius. Let me show you the Petri dish that I'm going to transfer them to. It doesn't have anything growing in it yet, but it has a layer of this yellow stuff called agar. Agar is like Jello. It has nutrients in it, and bacteria cells can live on the surface of agar and absorb the nutrients. And if you make the right agar with the right ingredients, the bacteria cells will thrive. The agar right now is sterile. It's completely sterilized because it was heated to a very high temperature in a machine called an autoclave. If I were to remove the lid right now and expose the agar to the air, like so, it could be contaminated by things in the air like mold. So I always keep the lid on. And when I want to open the dish, I use this technique. This is called clam shelling. I just crack it a little bit, and then I close it again right away. So I'm going to clam shell the lid and use my micropipette to transfer the bacteria cells, some of which are hopefully transformed, into the Petri dish. So I have a new sterile tip. I'm going to remove the bacteria from my green tube here. 250 microliters. This is going to go in the trash. I don't need that anymore. Here's my Petri dish. I'll clamshell, and then I just push the bacteria into a puddle on the surface of the agar. They're in like Flynn. Can I leave them in a puddle? I suppose I could. It looks a little crowded. So the next step is to move the puddle of cells all over the surface of the agar like icing on a cake. To do that, we use sterile glass beads. I'll show you. So in this tube, they look like Orbeez, are tiny little glass beads. They've been sterilized also in an autoclave. So they were heated to a very, very hot temperature, and any germs or microbes that might have been on them were killed. And they've been in this sealed tube ever since. I'm going to open the tube. And now I'm going to pour those beads into my Petri dish, and I'm going to move them around. I love that sound. So I'm moving the beads over the surface of the agar. They're spreading the cells all over, so they've all got a nice place to grow. And after about 10 seconds, I need to remove the beads. Can I use my fingers? No. What I need to do is tap the plate on the table or on my hand so all the beads collect in one corner, and then over my little trash beaker, I'll clamshell upside down and just knock the beads out. Ta-da! Now I cannot see any bacteria yet. In order to see the bacteria, I need to give them time to grow. Yeah, so what are they going to look like when they grow? Well, I can show you a picture, and I also have some real results. Here's a really fantastic picture. Fabulous results. So this is a Petri dish. that has colonies of growth. So the colonies are these little dots. There are a lot of colonies on this dish. This was a very efficient transformation. Lots of the cells took in new DNA. But you might see that there are some places where there's nothing growing. What happened in those places where there are no colonies? Well, don't forget, only a small % of the cells are transformed. What happens to all the cells that don't get plasmids inside them? They're eventually going to die. Their growth is going to be inhibited because the agar that we use in our experiment contains that antibiotic, ampicillin. So everywhere you see no growth, that's where the growth of the bacteria cells was inhibited by the antibiotic, and they eventually died. Here, where there's a colony, let's say we've got this colony right here, for example, or that one, it's by itself. That's where one cell landed on the Petri dish and survived because it was resistant to the ampicillin, and it started to divide. And overnight in the incubator, it formed a visible pile of cells. A colony is billions of cells, and that's why I can actually see it. So let's say my goal is to get lots and lots of green fluorescent protein. Can I get lots and lots of green fluorescent protein from a Petri dish like that? I could get some. Let's say I want more. Let's say I have some cells on a Petri dish that are making GFP for me, green fluorescent protein, but I want more glowing cells. Do I need to repeat my experiment? I don't. All I need to do is transfer some of these already transformed cells into a new Petri dish or into some liquid food and grow them again. They'll just keep reproducing, passing the gene on to their offspring, and they'll all be making green fluorescent protein for me. So imagine these cells were producing insulin, and I wanted more insulin producing cells. Same idea. I collect them from the Petri dish, and I continue to culture the cells. I continue to give them food and warmth until I have enough cells to get a batch of the protein that I want to collect. So what I'm going to do is show you how to transfer a colony of cells from a Petri dish into some liquid food so that you'll have a big batch of glowing cells to extract and purify the protein from. Now to do this, I'm going to have to turn out my lights. So give me a moment. What I'm going to do is show you a Petri dish that has fluorescent bacteria cells on it. And you're going to see that there's one little trick that I didn't discuss yet, and that's ultraviolet light. To see the bacteria glow, like in that picture, you have to turn out all of the bright white lights in the room and expose the bacteria to UV, or ultraviolet light. In a laboratory setting, to see bacteria glow that are producing GFP, they need to be exposed to blue light, and then they express the green light. Pretty neat. So this is a Petri dish of some pre-transformed cells. This is what the results from my experiment will look like tomorrow after they've had time to grow. And using a black light, I can identify a nice big glowing colony and transfer that colony into liquid food. The liquid food that we use is called LB broth, Loria Bertani, named after the scientist who created it. And it's like Campbell's soup for bacteria cells. It's what's in agar also. It just doesn't have the agar added to it, so it's still liquid. All I need to do is pluck one colony and put it in this liquid food, incubated overnight, and tomorrow the whole liquid, 2 milliliters of it, all of it will be full of glowing cells. And you'll see that in part 2. So what we're going to do is grow a batch of glowing cells so that a lot of green fluorescent protein can be extracted and purified. I'm going to turn the lights back on so that I can show you this last step. If you have any questions about what I've done so far or that I've discussed, please don't forget that there is a chat and there's a moderator in the chat to answer your questions. Okay, so I am going to use a pipette tip to do this, a sterile pipette tip. come back up to the camera. And I'm going to take the lid off my Petri dish all the way. See those colonies there? All I do is take a pipette tip and I lift one of those colonies. Boop. I just touched it. Now there's a little bit of bacteria on the end of my pipette tip. And all I need to do is remove the cap of my culture tube. So this is where the cells will grow. And I'm going to drop the pipette tip right into the liquid and cap it. And this is a loose fitting cap so that oxygen can get in. Overnight, this will be in an incubator that shakes the tube. So it keeps the cells warm and it aerates them. It keeps them moving around and they all have good oxygen. And overnight, the cells that are on that pipette tip will spread through the liquid and multiply. So for part 2 of this experiment, you'll be starting with an overnight culture of genetically engineered bacteria cells, all producing green fluorescent protein. And you'll learn how to extract the protein, remove it from those glowing bacteria cells, and purify it, separate it from all the other proteins inside the cell. And these are pretty much the same steps that would be done at a pharmaceutical company to batch produce insulin or another human hormone or enzyme to treat a human condition. I hope that you enjoyed my presentation today and learned a little bit about GFP. It was a pleasure to be here, and I look forward to seeing you again here at DNALC Live. Bye.